Have you ever wondered about the invisible instructions that make a living thing what it is? Think of a very, very long thread far too thin for our eyes to see. This thread is like DNA. It is a remarkable molecule found inside every living thing on our planet. From the tallest redwood tree in a silent forest to the smallest microbe drifting in the ocean currents, DNA is the blueprint. It carries all the information needed for life to grow, to function, to continue from one generation to the next. This thread holds the secrets of existence itself, a delicate and ancient code passed down through time. This thread of life is not just a simple string, it is a library of instructions. But what would happen if this incredibly long and important thread were to unravel completely? What if it unwound all the way like a ball of yarn tumbling down a long staircase, becoming a tangled mess? The clear answer is this. The DNA thread is astonishingly long. If you stretch DNA from one human cell, it would be about 2 meters. How does something so long fit inside a microscopic cell nucleus? The answer lies in masterful packaging. Instead, it winds the DNA very tightly around special proteins, much like winding thread onto countless tiny spools. This tightly wound package of DNA and protein is called a chromosome. This organization keeps the vast library of genetic information tidy and accessible. These chromosomes act as neat, manageable volumes in the genetic library. In humans, DNA is divided into 23 pairs of these chromosomes, for a total of 46. One set of 23 comes from the mother. The other set of 23 comes from the father, creating a unique individual. Other species have different numbers. A potato has 48 chromosomes. A fruit fly has only 8. Chromosome number does not determine complexity, but it is a species-specific characteristic, part of biological identity. You have likely heard of the X and Y chromosomes. They are a special pair called sex chromosomes, and they help determine biological sex in many animals, including humans. However, nature is diverse, and not all species use the XY system. Structure also provides protection and regulation. To understand what happens when DNA unwinds completely, we must first look at how it unwinds in a healthy, normal way. Think of the DNA thread as a zipper. When the cell needs to read a specific instruction, or gene, it does not unravel the entire chromosome. Instead, it carefully unzips a small section of the DNA double helix. This temporary opening allows the cell's machinery to access the code and make a copy of it. Once the instruction has been copied, the zipper closes back up. The DNA strand winds back into its stable helical shape, and the chromosome returns to its compact form. This controlled unwinding and rewinding is a fundamental part of life. The problem arises when the unwinding is not controlled. What if the thread unwound all the way and did not wind back? Or, even worse, what if the thread itself were to break? This is very different from the gentle unzipping needed to read a gene. A complete unwinding or a physical break in the DNA strand is a form of damage. The cell's machinery can no longer read them correctly. If DNA unwound all the way and broke apart, the cell would stop working and die. This is the simple and stark reality. When the DNA thread breaks or unwinds uncontrollably, the first and most immediate problem is that the genes become broken. A gene is a specific sequence of information, like a complete sentence that gives a clear instruction. If the DNA strand is severed in the middle of a gene, the sentence is cut in half. As a result, it fails to produce the correct protein that the gene was coding for. The cell is left without a critical worker it needs to perform its duties. Without the right proteins, the cell's functions begin to fail one by one. In a cell, proteins are needed to generate energy. In a cell, proteins are needed to transport materials. In a cell, proteins are needed to maintain its structure. If the proteins responsible for energy production are not made, the cell will run out of power. If the proteins that form its internal skeleton are missing, the cell will lose its shape and fall apart. Eventually, if enough essential functions are disrupted, the cell cannot survive. It will simply stop working and die. Sometimes the damage leads to a different kind of error. The cell might read the broken gene and produce a faulty, misshapen protein. This breakdown at the cellular level has profound effects on the entire organism. If growth control genes are broken, unchecked division can begin, the hallmark of cancer. Although the consequences of broken DNA are severe, life is remarkably resilient. Cells are not helpless victims of this damage. Every cell possesses an extraordinary toolkit of molecular machines dedicated to DNA repair. These are like tiny, vigilant maintenance crews that constantly patrol the DNA strands, looking for breaks, mismatches, other forms of damage. When they find a problem, they immediately go to work. These repair systems are incredibly effective, fixing thousands of DNA damage events in every one of your cells every single day. However, sometimes the damage is too extensive to fix. When a cell determines that its DNA is damaged beyond repair, it can activate a program of controlled self-destruction, apoptosis. This is not a chaotic death, but an orderly dismantling of the cell. It prevents broken, potentially dangerous cells from remaining. So if the DNA thread unwound completely and broke, the cell would fail, and the living thing could die. This remains the simple, direct answer. But the sophistication of DNA repair offers a message of hope, helping us treat disease and understand the resilience woven into the very thread of life.